Very good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are in our 100 days challenge topics for NEET 2026, of which yesterday we have begun with plant physiology of photosynthesis in higher plants, wherein so far we have completed early discoveries and all the three sequential events of light reaction, and we have also dealt the non-cyclic photophosphorylation as well. Today we are going to discuss about chemiosmotic hypothesis on how ATP is formed in non-cyclic photophosphorylation. After non-cyclic photophosphorylation in higher plants of oxygenic photosynthesis, how ATP is being formed, we learn through chemiosmotic hypothesis. And when it comes to light independent reactions, today we are going to see C3 cycle, photorespiration, which is operated only in C3 plants, and we are also going to look into C4 cycle, which is also called as Hutchinson-Slack pathway. First, let us begin with chemiosmotic hypothesis. Chemiosmotic hypothesis beta. So, this is given by Peter Mitchell. Okay, chemiosmotic hypothesis is given by Peter Mitchell. Fine. So, this chemiosmotic hypothesis suggests that how ATP is being formed. To know how ATP is being formed by the electrochemical gradient here, first you have to remember the three events which happens in the non-cyclic photophosphorylation. First is photolysis of water. The very first reaction is what ma? Photolysis of water, which happens through oxygen evolving complex, which is located on the inner membrane of the thylakoid towards lumen. This will be releasing out protons. This proton concentration will be more in the thylakoid lumen. This is clear to you, right? This is the first point which is saying you that proton accumulation is taking place inside the thylakoid lumen. This is because of what ma? Photolysis of water. Second point is the protons are the primary acceptor of the electron here. Protons are accumulating in the lumen, and two electrons which are released as a result of photolysis of water, those two electrons are submitted to the reaction center and accepted by the primary acceptor. In your NCERT there is a clear line beta. The primary acceptor here is pheophytin. Pheophytin is the primary electron acceptor here. That pheophytin is located towards the outer side of the thylakoid membrane, and the electrons are given to plastoquinone. This plastoquinone is not just an electron carrier, but this parallelly accepts the protons from the stroma as well, and this delivers those protons to where? To the lumen of the thylakoid. Okay. So, here these 2 points are clear to you na beta. After photolysis of water, which is in the thylakoid lumen, very good morning ma, which is in the thylakoid lumen, here protons get accumulated in the lumen and electrons are submitted to where? Electrons are submitted to the primary electron acceptor, that is pheophytin. Pheophytin is located on the outer side of the thylakoid membrane, and then the electrons are submitted to plastoquinone. Plastoquinone is not just an electron acceptor, but it is also a proton acceptor. That proton acceptor is from stroma. Protons from stroma are being delivered to the lumen of the thylakoid. 2 points are clear to you, right? Third one, the final or the ultimate electron acceptor is NADP plus. Okay, the ultimate or final electron acceptor is what ma? NADP plus. Here this NADP plus accepting protons which are present in the stroma gets reduced to NADPH. Okay, and this is an enzyme catalyzed reaction, and the enzyme over here is what ma? NADP reductase. The enzyme over here is NADP reductase. So, here 3 events you have to remember. First, that is proton accumulation because of photolysis of water, and second, electrons which are given to the primary acceptor, that is pheophytin, located on the outer side of the thylakoid membrane, which is giving the electrons not just to an electron carrier but also a proton carrier, that is plastoquinone. Plastoquinone is located towards the inner side of the thylakoid membrane. This parallelly accepts protons from the stroma and delivers it to the lumen of the thylakoid. This is one reason where protons are getting reduced in the stroma and increasing further in the lumen. And third event which is reducing the proton concentration in stroma is the last step, wherein NADP accepting the electrons, okay, NADP, which is the finer electron acceptor of ETS, this gets converted to NADPH by the enzyme NADP reductase. Okay, this is by the enzyme NADP reductase, which gets reduced to NADPH. This utilizes the protons of the stroma. Because of these three events, the proton concentration in the stroma will be low and the proton concentration in the thylakoid lumen will be increased. As such, the proton gradient is generated in between the thylakoid lumen and the stroma, because of which ATP synthase pump will be activated. Understood, ma? Because of these three events, proton pump is generated, that is from the lumen to the stroma. Proton pump is generated from the lumen to stroma. Remember here, more protons are present in the lumen, thereby less pH is there in the lumen, and less protons are present in the stroma, thereby more pH will be there in the stroma. Remember that, ma. We may get statement based questions as such also. So remember each and every event and how you can apply based on the question. Application is more important here. Okay. So here as such, because of the proton gradient, proton pump is generated from lumen to the stroma. Okay. So here in this proton pump we have F0, F1 channel. F0 is embedded inside the thylakoid membrane and F1 is towards the stroma. In a single turn, it will release 4 protons, which helps or aids in the phosphorylation of ADP and inorganic phosphate. Okay. Phosphorylating this forms out what? ATP. Okay. This leads to the formation of ATP. Clear, right, beta? Complete chemiosmotic hypothesis. And here there is one more major point to remember is, what are the requirements or what does chemiosmotic hypothesis require or this process requires? First is a membrane, that is thylakoid membrane, and this chemiosmosis happens both in the chloroplast and in the mitochondria as well. Okay, chemiosmosis happens both in the chloroplast and mitochondria. In chloroplast though it is from lumen to stroma, in mitochondria though it is from intermembrane space to the mitochondrial matrix. Remember beta. So here it requires a membrane. Second is a proton gradient, and third is proton pump, and fourth will be ATP synthase. This is directly from your NCERT. You have to remember this for sure. So whatever we have discussed from chemiosmotic hypothesis is very important beta. If you want, you can take a screenshot of this also. Next. So here is an elaborate description about ATP synthase. This ATP synthase is having two parts, that is F0, F1 channels or F0, F1 pumps. F0 is embedded inside the thylakoid membrane as we have discussed, and this forms a transmembrane channel that carries the facilitated diffusion of H plus ions or protons from where? Lumen to stroma across the membrane. What is it carrying out, ma? Protons. Through what diffusion? Facilitated diffusion. What is facilitated diffusion? Transfer of any molecules either through a carrier protein or through a channel protein is said to be what? Facilitated diffusion. Next here F1 pump, this is towards the stroma. This protrudes the outer surface of the thylakoid membrane, this faces the stroma. Breakdown of the gradient provides enough energy to cause a conformational change in the F1 pump, which makes the enzyme synthesize packed ATP. Clear, right, ma? This is a description about F0, F1 pump. Next. Let us begin with the dark reactions. I already told you light independent reactions or dark reactions. Calling dark reactions light independent reactions. Look at here. Light independent reactions. Calling light independent reactions as dark reactions is a misnomer, because it doesn't mean that it takes place during night or dark conditions. Light independent reactions only means that they are not directly dependent on light, they are dependent on products of light reaction, those are ATP and NADPH. Okay, products of light reaction, those are ATP and NADPH. Clear, right, ma? So here if you see C3 cycle, C4 cycle and CAM cycle, in different ways it will be operated in different plants. As such, the carbon dioxide is fixed to glucose. C6H12O6, it will be fixed to what? Glucose. So C3 cycle, which is also called as Kelvin cycle, because this is discovered by Melvin Kelvin by conducting algal photosynthesis. By conducting what, ma? Algal photosynthesis. He has taken a carbon isotope of C14 and worked it out on chlorella and conducted oxygenic photosynthesis, whereby he said that the first stable organic intermediate formed is 3-phosphoglyceric acid. The first stable organic intermediate formed is what, ma? 3-phosphoglyceric acid, that is 3-carbon organic acid. This occurs in all the photosynthetic plants. Clear, right, ma? So C3 cycle is operated in all the photosynthetic plants, specifically in C3 plants. If we have to say, C3 plants are temperate plants, beta, do remember. C3 plants are temperate plants which grow in low temperatures. At low temperatures, there will be high carbon dioxide to oxygen ratio. Remember till here, okay? It is discovered by Melvin Kelvin by conducting algal photosynthesis using radioactive isotope, that is C14, worked it out on chlorella, and he said that the first stable organic intermediate is what? 3-phosphoglyceric acid. Okay, this occurs in all the photosynthetic plants. C4 cycle, which is also called as Hutchin-Slack pathway. Here the first stable organic intermediate formed is oxaloacetic acid, which is a 4-carbon compound. That is why we name it as what? C4 cycle. Later, oxaloacetic acid can be turned to malic acid or aspartic acid, which is again 4-carbon. This occurs only in C4 plants. What are C4 plants here? C4 plants are tropical plants. C3 plants are temperate plants. C4 plants are tropical plants. Most of the monocots here like sorghum, maize, sugarcane, this will conduct what? C4 cycle, as they are tropical plants. Remember with a simple mnemonic like SMS, sorghum, maize and sugarcane. These are most repeatedly asked in the NEET, okay? Considering these examples only, they will be asking most of the time, that is sorghum, maize, sugarcane and some millets. Here only dicot which conducts a C4 cycle is what, ma? Amaranthus. Amaranthus is the only dicot which conducts C4 cycle. And next is what? CAM cycle. CAM cycle is described as crustacean acid metabolism, which is the first stable organic intermediate, which is oxaloacetic acid. This occurs in all crustacean family members. Okay, this occurs where, ma? In all crustacean family members, and this plants are called as what? Desert plants. CAM cycle is activated or operated in desert plants, or succulent plants also you can call it as. Best examples can be cactus, agave, opuntia and aloe vera, and few tropical epiphytes like what? Orchids. This is also a good example. So you have to remember all these points, ma. Whatever I have discussed are very important for your NEET. First, let us look into the mechanism of C3 cycle. We will finish off C3 cycle first. So C3 cycle, if you have to consider, C3 cycle, as I told you, this is operated in all the plants, and this is operated in which cells? Mesophyll cells. Mesophyll cells. and stroma of the chloroplast. Remember the locations exactly. Stroma of the chloroplast, wherein C3 cycle has three major steps. One is carboxylation, second is reduction, and third is regeneration. Carboxylation, reduction and regeneration. So, here if you see beta, first carboxylation, carbon dioxide whenever it is being entered into the mesophyll cell or stroma, this carbon dioxide is primarily accepted by what? 5 carbon compound, that is RUBP. In C3 cycle, what is the primary acceptor of carbon dioxide? If that is the question, primary acceptor is what ma? RUBP. This is catalyzed by the world's abundant enzyme, that is rubisco. Ribulose bisphosphate carboxylase oxygenase. This rubisco has equal potential to bind both with carbon dioxide and with oxygen. Here, after this reaction, the primary intermediate or the primary product that is formed is 3-phosphoglyceric acid. This 3-phosphoglyceric acid, 2 molecules of 3-phosphoglyceric acid are formed, 3 carbon each. So, 1 carbon dioxide accepted by the primary carbon dioxide acceptor of C3 cycle, that is RUBP, catalyzed by rubisco, forms the primary product, that is 3-phosphoglyceric acid, 2 molecules of 3-PGA. Clear, right ma? So, here 1 plus 5 total 6 carbons and the product will be 2 molecules of 3 carbon compound. Remember as such. Next step is what? Reduction, wherein 2 molecules of phosphoglyceric acid will be reduced to 2 molecules of glyceraldehyde 3-phosphate. For this reduction step, for every carbon dioxide molecule, it requires 2 ATP and 2 NADPH. Do remember ma, if arrow is like this, then it is utilized in the reaction. If arrow is like this, then it is released out by the reaction. Mostly this you will see in the respiratory pathways, this you will see in the photosynthetic dark reaction pathways, wherein you see here in the reduction step 2 ATP and 2 NADPH are utilized, which are converted to 2 ADP and 2 NADP plus. Here 2 molecules of glyceraldehyde 3-phosphate are regenerating 1 molecule of RUBP. So, here 2 molecules of G3P means how many carbons beta? 6 carbons. From the 6 carbons, 5 carbon RUBP will be regenerated back. Rest 1 carbon will be leaving the cycle all the time to form the glucose chain. Okay. So, from 1 carbon dioxide continuing the cycle, okay, 1 carbon dioxide, 1 carbon will be releasing to form the glucose chain. As such, 6 cycles will release how much? How many carbons? 6 carbons. 6 carbons will make 1 glucose molecule. So, here to form 1 glucose molecule, how many carbon dioxide fixations have to take place ma? 6 carbon dioxide fixations, and how many times Kelvin cycle has to run? 6 times Kelvin cycle has to run. Clear, right? Understood till here? So, here carboxylation in reduction for every carbon dioxide 2 ATP and 2 NADH are utilized, and from this 6 carbon, that is 2 molecules of G3P, 1 RUBP will be regenerated, the rest 1 carbon will be leaving the cycle all the time to form what? Glucose molecule, which is 6 carbon glucose. As such, to form 1 glucose molecule, 6 carbon dioxide fixations, 6 cycles have to happen. Yes. This is clearly describing you out. You can see this diagram, and whereby we will be exercising the energetics as well beta. So, here in our previous this thing we have discussed about 1 carbon dioxide running 1 Kelvin cycle. Here 6 carbon dioxide molecules to form 1 glucose chain. Can you see this? So, here let me tell you what are the energetics for 1 carbon dioxide fixation or 1 Kelvin cycle. For 1 Kelvin cycle, how many carbon dioxides involved ma? 1 carbon dioxide. How many ATP molecules are utilized? 2 in the reduction step and 1 will be in the regeneration step. So totally 3 ATP molecules will be utilized in the reaction. 3 ATP will be utilized in the reaction. And how many NADPH? 2 NADPH will be utilized. 2 NADPH will be utilized in the reaction. Clear, right, ma? Next, for 6 carbon dioxide or to the formation of 1 glucose molecule, here 6 times Kelvin cycle have to run, right? 6 Kelvin cycles. How many carbon dioxide fixations? 6 carbon dioxide fixations. And how much ATP? 6 into 3. For 1 carbon dioxide, if it is 3 ATP, for 6 carbon dioxides, it is how much, beta? 6 into 3, that is 18 ATP, right, ma? Next, for 6 molecules of carbon dioxide or 6 Kelvin cycles, how much NADPH is required? That is, for 1 carbon dioxide, it is 2 NADPH. For 6 into 2, it will be 12 NADPH. Clear, right? So here completely for 1 carbon dioxide and for 6 carbon dioxides, I have given you out the energetics. This is very much important for your NEET. Okay, and this is for 1 glucose molecule, ma. This is for 1 glucose molecule, right? So along with this, let us also discuss the photorespiration. So today we will be ending up with photorespiration. As we have discussed about the Rubisco action, Rubisco has dual nature, that is ribulose bisphosphate, carboxylase and oxygenase. Carboxylase and oxygenase, right, ma'am? So this ribulose bisphosphate, carboxylase, oxygenase, it has equal potential to bind with carbon dioxide and oxygen. So at low temperatures, high carbon dioxide and oxygen ratio will be there. At high temperatures, low carbon dioxide oxygen ratio will be there in the atmosphere and in the intracellular spaces of the leaf cells as well. So here at low temperature, when high carbon dioxide and oxygen ratio is there, Kelvin cycle is operated usually, because there is high carbon dioxide and oxygen. Definitely Rubisco will bind with carbon dioxide only, and that will run what? Kelvin cycle. But when there is high temperature, the solubility, carbon dioxide will be easily solubilized. Okay, carbon dioxide will be easily solubilized, so that this Rubisco, instead of binding with carbon dioxide, it binds with what? Oxygen. This binds with oxygen and starts up photorespiration or C2 cycle. So here what's at high relative concentration of oxygen, high temperature, Rubisco behaves as what? Oxygenase. Based on the atmosphere, its behavior changes. Phosphoglycolate is formed, which inhibits the steps of Kelvin cycle. ATP is consumed, carbon dioxide is released, and no formation of glucose. That is why photorespiration is said to be a wasteful process that is operated only in C3 plants, because C3 plants doesn't have any alternative other than C3 cycle, which is in the mesophyll cells. So let us look into photorespiration clearly. First point you have to remember is in which organelles photorespiration is operated. Photorespiration is operated in 3 organelles, ma. First, that is chloroplast. Second will be peroxisomes. Third is mitochondria. Okay. So what are the 3 organelles that are involved in photorespiration? The 3 organelles that are involved in photorespiration are chloroplast, peroxisomes and mitochondria. So here you see beta, in chloroplast, when one molecule of what happens, firstly RUBP along with oxygen, right? Here there is no carbon dioxide, because carbon dioxide fixation is not happening. Rubisco is behaving as what? Oxygenase over here. So here 5 carbons are there on the reactant side. So only 5 carbons will be formed in the product side. As such, what are the products formed? One 3-carbon compound, that is phosphoglyceric acid, and one 2-carbon compound, that is phosphoglycolate. Okay. So, here in the photorespiration from the primary step, that is RUBP along with oxygen, rubisco behaving as oxygenase, this is forming out what? One molecule of 3 carbon compound, that is phosphoglyceric acid, and one molecule of 2 carbon compound, that is phosphoglycolate. Only one molecule of phosphoglyceric acid cannot operate the Kelvin cycle, right ma? 2 molecules are required. So, to generate one more molecule of PGA, it has to travel all across these organelles, and all across this energy will be utilized. So, how this one molecule of phosphoglycolate will be first undergoing dephosphorylation, turning out to form glycolate. Glycolate will be entering the peroxisome, turns to glyoxylate, wherein it utilizes oxygen over here, and glyoxylate turns out to be glycine. Glycine amino acid will be entering mitochondria. In mitochondria, carbon dioxide is released. So, have you seen ma? First oxygen is utilized in the peroxisome, carbon dioxide is released in the mitochondria. Along with carbon dioxide, ammonia is also released. NADH will be released over here. Next, this glycine is converted to what? Serine. Serine is again entering to peroxisome, converted to hydroxypyruvate, then glycerate. This hydroxypyruvate to glycerate, NADH is utilized. So, it is balanced. Here it is released, here it is utilized. So, there is no energy driven over here. Glycerate entering to chloroplast again, then it will be synthesizing phosphoglyceric acid utilizing one ATP. Okay. So, here ATP is utilized, oxygen is utilized, carbon dioxide is released, there is no sugar synthesis only. That is why photorespiration is said to be a wasteful process, okay, which is operated only in the C3 plants, right ma. So, this photorespiration is operated only in the C3 plants when there is high temperature, low carbon dioxide oxygen concentration or ratio. Clear till here ma? And in tomorrow's class, we will be exercising C4 cycle and CAM cycle together beta. So, till here it is enough today, and I would like to end the session by giving you a brief introduction on NEET World's special programs, wherein we have 3 special programs for our NEET World. First is NEET World's 2 steps to medicine with a minimal fee of 3333 per month. NEET World's 1 step to medicine with a fee of again the same 3333 per month. NEET World's power step to medicine for long term students or repeaters with the same fee 3333 per month. So, any of these programs can be utilized for any students, you know, as this is feasible economically, it is feasible to all the students of different financial backgrounds. So, this will be helpful for most of the students. To get into these courses or to know more about these programs, you can call us to the given number, or you can directly visit our branch which is in Narayanguda, Hyderabad. And finally, to all the students who are studying in the public platform or you are doing self study for NEET 2026, the NEET World is here with you, that you can log into our NEET World's Telegram group, wherein daily quizzes of biology, chemistry, physics and maths who are also preparing for JEE mains. This daily quizzes will be conducted, live class schedules will be updated, and free resources are provided. Free resources of study materials, question banks will be provided from our NEET World, and one-on-one doubt solving sessions with faculties are directly facilitated through the Telegram group. So, whoever students are interested, you can join our Telegram group directly by scanning this QR code or by visiting our official website, that is www.neetworld.in. Okay, so with this I will sum up today's class, and we will meet tomorrow, and we will be discussing C4 cycle and CAM cycle. Attend that without fail. Thank you.